Welcome back to the Deep Dive. We are digging into the fine print of your biology today. We're focusing on a molecule that's, well, tiny, really simple on the surface. And yet, it represents one of the most, I guess, frustratingly complex and important puzzles in modern neuroscience. We're talking about DSIP.
That's right, DSIP, the delta sleep-inducing peptide. And it really is a molecule just shrouded in contradiction.
How so?
Well, for decades now, we've seen DSIP, or something very much like it, regulating some of your body's most fundamental operations. We're talking deep sleep, hormonal stability, even stress resilience. It acts like this quiet, really crucial operator in the background.
Okay, sounds vital. So what's the contradiction?
The paradox is this. Scientists still haven't found the blueprint. We don't know how it's made exactly. There's no specific gene identified for it. And we also haven't found the precise dock it uses, the specific receptor to deliver its commands. It's a bit of a ghost in the machine.
Wow, okay. So that's the central conflict we're diving into today. We have this molecule. It promotes deep restorative sleep. It helps stabilize major hormone systems.
Mm-hmm.
But the gene that codes for it and the receptor that catches it, they're just missing from the map.
Essentially, yes, that's the puzzle.
All right, so our mission today is to extract the concrete facts we do know about this mysterious nonapeptide. We want to explore its surprisingly broad roles way beyond just making you sleepy and really highlight the biggest unresolved questions that define, well, the current frontier of resilience science. Okay, let's unpack this system, starting right at the basics. What exactly is DSIP?
Structurally speaking, DSIP is remarkably straightforward, which is honestly part of its elegance. It's classified as a nonapeptide, meaning it's made up of just 9 amino acids strung together, a very short chain. We even know the exact sequence. Tryptophan, alanine, glycine, aspartic acid, alanine, serine, glycine, glutamic acid, or alisergly for short.
Okay, hearing that list, it still sounds a bit complex, but the fact that it's only 9 amino acids, that's significant, right, compared to other signaling molecules.
Oh, critically important, yes. Its molecular weight is tiny, only about 850 daltons.
And for comparison?
Well, most typical proteins, many neurohormones, they weigh thousands, sometimes even tens of thousands of daltons. So DSIP is really small fry in terms of size.
And does that small size matter for what it does?
Absolutely. It's key because it likely contributes to its ability to traverse biological obstacles, to get around the body.
Okay. And looking at that chemical sequence you listed, is there anything special about the specific amino acids?
Yeah.
About the composition that allows it this sort of freedom of movement?
Yes, definitely. The peptide has parts that are hydrophobic, water-hating regions, and parts that are hydrophilic, water-loving regions.
So it can handle different environments.
Exactly. This dual nature makes it amphiphilic. That means it can interact comfortably in diverse biological settings. It can travel through the watery medium of, say, blood plasma, but potentially also slip through the lipid-heavy fatty membranes, like the blood-brain barrier.
Which is notoriously difficult to cross.
Very difficult. So the small size, combined with this dual chemical nature, it means DSIP could potentially influence both the central nervous system, the brain, and peripheral organs with, well, surprising ease.
That gives it access to pretty much the entire body then. Okay, let's rewind a bit. You mentioned decades of research. Let's go back to the 1970s. Tell us the origin story. How did we even first detect this tiny traveling operator?
Yeah, the discovery, it's a classic piece of early neuroscience detective work. Scientists, particularly a group in Switzerland, they hypothesized that sleep wasn't just a passive switching off.
Right, not just the absence of wakefulness.
Exactly. They thought maybe something active was circulating in the blood of a sleeping animal, something that could actually induce sleep in another animal.
So they were literally looking for a circulating factor like contagious sleep in a molecule.
Precisely that, yeah. They used a technique called cross-perfusion. It sounds a bit Frankensteinish now, but
How did it work?
they would take blood specifically, the venous effluent, so the blood leaving the brain from rabbits that were deep in slow wave sleep, that deep delta sleep stage.
Okay.
Then they extracted a sort of crude fraction from this blood. They purified it somewhat. And then they infused this extract into the ventricular system, basically, into the fluid spaces of a wakeful recipient rabbit.
And the results? What happened to the awake rabbit?
The recipients showed an immediate, quite observable increase in the specific electrical brain activity associated with deep restorative rest, those delta waves.
Oh, hence the name.
Hence the name. This led them eventually to isolate that specific 9 amino acid structure, which they called the delta sleep-inducing peptide, or DSIP. The discovery itself was, well, monumental at the time. It was proof that sleep could be actively regulated by a humoral factor, something circulating in the body fluids.
That really sounds like a breakthrough moment, an aha, that should have solved the sleep puzzle.
Yeah.
But, as you said, this is where the mystery really thickens, the question of biosynthesis. If it causes sleep, if it stabilizes systems, the body has to be making it deliberately somehow. So, where's the blueprint?
And this is arguably the biggest enigma that still plagues DSIP research today. It's quite frustrating. We have the exact chemical formula, we know what it looks like, and we can detect DSIP-like immunoreactivity, basically signs of its presence in crucial neurological centers.
More specifically.
Places like the hypothalamus, the limbic system, the pituitary gland, key areas for regulation, and even in peripheral organs like the pancreas and the adrenals. But despite all that,
still haven't found the gene.
we have never identified a unique gene in the human genome, or other mammalian genomes for that matter, that specifically codes for DSIP.
Wait, that's genuinely astonishing. In modern molecular biology, the usual path is find the gene, find the protein it codes for. This feels like finding a really complex, perfectly functional key, confirming it unlocks multiple important doors in the house, but then realizing the factory blueprint for that specific key just isn't in the architectural plans anywhere.
It is baffling. And it forces us to confront some pretty unconventional theories about how it's produced. We're left with, broadly speaking, 2 main hypotheses for how DSI actually exists in the body.
Okay, what's the first one?
The first theory suggests DSIP isn't a primary product itself. It's not made from scratch off its own gene template. Instead, it might be a byproduct.
A byproduct of what?
It might form from a much larger, more common, maybe even ubiquitous protein, a protein that performs a completely different job most of the time.
So the idea is that the body takes this other larger protein and snips out just this 9 amino acid sequence under certain conditions.
Exactly. It would involve cleaving or breaking down this larger precursor protein only at the very specific points that release the DSIP sequence. This is known as post-translational modification. It happens after the main protein is already made.
And what would trigger this snipping process?
Well, the enzyme responsible for the cleavage would likely only be activated under very specific physiological demands, perhaps periods of high stress, maybe extreme fatigue, or maybe at the onset of the deep sleep phase itself.
Why would evolution choose such a complex, sort of hidden way to make it? Why not just have a dedicated DSIP gene? Seems simpler.
Well, thinking about it evolutionarily, this model actually offers potentially immense regulatory flexibility and speed.
Speed, how so?
If DSIP is needed right now, say, to buffer an acute stressor or initiate recovery processes quickly producing it by snipping an existing, abundant protein is much faster than going through the whole process of transcribing a gene from DNA, translating the mRNA into protein, and then folding that new protein correctly.
Ah, I see. It's like having the raw materials already on the shelves, ready for rapid assembly when the alarm bell rings.
Exactly, a rapid response manufacturing process. Now, the second major hypothesis tries to address its stability, or rather, its lack of stability.
Okay.
Given its simple structure, just 9 amino acids, and its vulnerability to enzymes called peptidases, which are everywhere in the body and chew up peptides, DSIP naturally has a very short half-life. It doesn't last long on its own.
So it breaks down really quickly after it's made.
Very quickly, which leads researchers to theorize that once it is cleaved from its precursor, maybe it has to immediately bind to something else, like carrier proteins, perhaps, in the blood and the cerebrospinal fluid.
And these carrier proteins would protect it.
Exactly. They would shield the little nonapeptide, stabilize it, protect it from those degrading enzymes. This would allow it to travel longer distances through the body and perform its function before eventually being cleared from the system.
So the production process is still a bit of a black box, but the theories point towards a system designed for really rapid, context-dependent recovery signals.
That context seems absolutely vital, yes. We're dealing with a molecule that seems intrinsically tied to the moment of recovery, maybe produced only when it's truly needed.
Okay, that makes sense. Let's move then from the mystery of its origin to the function that actually gave it its name, its primary role, or at least initially perceived role, in sleep regulation.
Right. And the initial findings back in the 70s and 80s gave everyone a lot of hope for maybe, a perfect natural sleep aid.
Because the animal data was strong.
Very strong. Those early animal experiments firmly supported the name. They showed DSIP increased delta EEG activity, promoting that deep, slow-wave sleep, which we know is essential for physical restoration, memory consolidation, all that good stuff.
And when they tried to translate that to humans, did those results hold up consistently?
Well, in some small early human trials, we definitely saw encouraging signs. Participants often reported a reduction in sleep latency.
So falling asleep faster.
Falling asleep faster, yes. And also measurable improvements in sleep efficiency, meaning they spent less time awake during the night, fewer interruptions.
Okay, that sounds fantastic on the surface, but let's go back to the point we raised earlier. If this was a totally consistent, reliable sleep switch, surely it would be a major pharmaceutical product by now, wouldn't it? The inconsistency you mentioned suggests there's a major nuance here. Something that separates DSIP from, say, a traditional sleeping pill.
That is absolutely the critical distinction we need to emphasize for you, the listener. DSIP is not a pharmaceutical hammer. It's not Ambien. The results across larger, more diverse groups of people are notoriously inconsistent.
So it doesn't work for everyone, or not all the time.
Exactly. Its efficacy is generally described as mild, maybe subtle, and it seems heavily dependent on both external conditions like your stress levels, the time of day it's given, and internal conditions like an individual's specific sleep patterns or problems.
So, if the animal data was so clear, showing that delta weight increase, but the human data is so variable, should we be a bit skeptical? Is it possible DSIP isn't actually the main driver of sleep, but maybe just an artifact, like a biomarker that happens to show up when deep sleep starts, rather than causing it?
That's a fair scientific challenge, and it's been debated. The data we have suggests it's probably more than just an artifact, but definitely less than a primary trigger, like a conventional hypnotic drug.
How so?
Well, consider the mechanism of a typical pharmaceutical hypnotic. It often works by, say, flooding your GABA receptors, essentially forcing your brain activity towards inhibition, towards unconsciousness. It slams the system into sleep mode.
Right.
DSIP doesn't seem to do that at all. If you're extremely stressed or highly anxious, your system is flooded with alerting neurotransmitters, stress hormones like cortisol. The relatively mild regulating effect of DSIP might just be completely overwhelmed by that cascade. It can't shout louder than the alarm bells.
Okay, so if I take a traditional sleeping pill, it's like a hammer forcing the door to my consciousness shut. DSIP is maybe more like oiling the hinges on the door, making it easier to close smoothly when conditions are right. But if the house is on fire behind the door, just oiling the hinges isn't going to make much difference.
That's actually a perfect analogy, yes. DSIP seems to function more as a regulator or maybe a facilitator. It helps optimize the body's own inherent sleep mechanisms. It seems to promote deeper, more efficient sleep by subtly modulating the internal environment in which sleep occurs. It helps ensure the body's natural rhythm, its circadian clock, is running smoothly and efficiently, but it probably can't override massive internal disruptions or external pressures.
Okay, that shift in perspective, seeing it less as a sleep trigger and more as a systemic regulator, that makes a lot more sense, especially as we move beyond just the bedroom into Section 3. DSIP potentially acting as this broader system stabilizer, particularly in times of stress.
Indeed. And what's really fascinating here is how DSIP's function seems to extend deep into the endocrine system, the neuroendocrine system. This strongly suggests its core biological mission isn't just about making you drowsy. It's more about restoring overall physiological homeostasis.
Homeostasis, that internal balance.
Exactly, that physiological sweet spot, especially after periods of, fatigue, intense physical exertion, or acute stress. It seems to be about recovery.
Let's talk about that stress buffer function specifically then, because stress is inherently destabilizing for the body, right? How does this tiny peptide apparently step in to help calm the system down?
It seems to engage directly with the core stress axis, what we call the HPA axis. That's the hypothalamic pituitary adrenal axis.
Right, the pathway that leads to cortisol release.
Precisely. When you encounter a stressor, your hypothalamus releases CRH, corticotropin-releasing hormone. That tells your pituitary gland to release ACTH, adrenocorticotropic hormone. And that hormone, ACTH, travels to your adrenal glands and tells them to flood your system with cortisol, the main stress hormone.
Okay, the classic stress response cascade. Where does DSIP fit in?
Evidence suggests that DSIP can actually reduce the levels of circulating ACTH.
So it intervenes quite early in that HPA cascade. It's like lowering the volume on the body's main alarm signal before it gets too loud.
Exactly like that. By potentially blunting that ACTH signal from the pituitary, DSIP consequently reduces the amount of cortisol that gets released by the adrenals. In effect, it acts almost like a neuroprotective shield, buffering the body against some of the severe physiological fallout that can come from prolonged or very acute stress. It seems to be a key part of the body's natural wind-down and recovery process after stress.
That's a potentially massive regulatory role for such a small molecule.
Mm.
But the hormonal interactions don't just stop at stress, do they? You mentioned growth and reproductive hormones earlier as well.
That's right, yes. There's also evidence that DSIP can stimulate the release of both growth hormone GH and luteinizing hormone, or LH.
GH and LH. Okay, what's the significance of that?
Well, GH is crucial for tissue repair, growth, metabolism, essentially physical restoration. LH is key for reproductive functions. Stimulating these further solidifies DSIP's potential role as a molecule involved in deep repair and restoration, perhaps coordinating these processes to occur optimally during the rest period.
Okay, how does it manage to stimulate growth hormone release? Does it just send a direct release GH signal to the pituitary?
It seems to employ a more sophisticated, kind of multifaceted approach. Yes, DSIP appears to stimulate GH release, but critically, it seems to achieve this, at least in part, by reducing the activity of another hormone called somatostatin.
And somatostatin does what?
Somatostatin is sometimes known as the growth hormone inhibiting hormone. Its job is basically to put the brakes on GH release.
Ah, I see. So DSIP isn't just pushing the accelerator for growth hormone, it's simultaneously taking the foot off the brake by inhibiting the inhibitor. That's quite clever coordination.
It suggests a really integrated regulation, doesn't it? It ensures that the growth and repair processes can happen efficiently during that restorative state facilitated by DSIP. And this stabilizing effect seems to extend even further to immediate vital functions like the cardiovascular system.
How so?
Some research suggests that DSIP may help to stabilize blood pressure and perhaps overall cardiac function, particularly when the organism is subjected to stressful or physiologically demanding conditions.
Okay, so if we try to zoom out and look at the whole picture then, we've got sleep regulation, stress buffering via the HPA axis, stimulation of restorative hormones like GH, possibly cardiac stability. It really starts to sound like DSIP could be functioning as this comprehensive system-wide reset button, something that activates the body's recovery protocol when it senses things are overworked or out of equilibrium.
I think that's a very fitting description. Yes, a reset button, or maybe a system maintenance coordinator. It seems to ensure that the body uses the opportunity of rest, perhaps deep sleep especially, to conduct full system maintenance and repair rather than just passively waiting for the next bout of activity or stress.
Which brings us right back to the core molecular puzzle, the ghost in the machine you mentioned earlier. If DSIP is acting as this highly effective background technician, telling the stress system to quiet down, telling the repair systems to gear up, how is it actually sending these complex instructions without its own dedicated phone line, so to speak? Section 4 is all about this mechanism of action, or rather, the enigma defined by that missing receptor.
This really is the scientist's headache with DSIP. As we said, in decades of pretty rigorous molecular study, we have never definitively confirmed a specific dedicated DSIP receptor. Normally, a peptide or hormone is the key, and the receptor on the cell surface is the lock. DSIP is clearly a key. We see its effects, but we just can't seem to find the unique lock it's supposed to fit into to deliver its primary message. It's incredibly elusive.
So if it doesn't have its own specific receptor, how does it communicate with the cell at all? Is it just sort of floating around nearby and having subtle physical effects, maybe changing membrane fluidity or something? Or is it actively engaging with the cell's machinery in some other way?
It seems to be the latter. It is actively engaging, but it appears to do so through more indirect methods. The current scientific consensus is leaning towards DSIP functioning primarily through modulation rather than direct primary messaging.
Modulation versus messaging. Can you break that down?
Sure, think of, say, insulin. Insulin binds its receptor, and that sends a very clear primary directive: open the gates and let glucose in. That's messaging. DSIP seems to be different. It's maybe more like an IT specialist who comes in and optimizes the existing network. It doesn't send the main email, but it makes sure the servers are running smoothly, the bandwidth is good, so that other messages get delivered faster and more efficiently. It seems to fine-tune the signals that are already being sent by other neurotransmitters and hormone systems.
That's a really helpful distinction. Okay, so what specific networks or systems does it seem to be fine-tuning or modulating?
Well, researchers have confirmed some quite complex interactions with several critical components of neural signaling. For instance, there's evidence it interacts with NMDA receptors.
NMDA receptors.
Yeah,
Those are crucial for learning and memory, right? Synaptic plasticity.
absolutely vital, yes. Changes in NMDA receptor function can have profound effects on brain excitability and long-term changes in neural connections. DSIP also seems to engage with adrenergic receptors.
Adrenergic.
Hmm.
Related to adrenaline, the fight-or-flight system.
Exactly. Adrenergic receptors are central to the sympathetic nervous system response. So DSIP interacting there connects very neatly back to the stress buffering role we discussed. If it's somehow dialing down the sensitivity or activity of those adrenergic receptors, it could help the system transition away from a high alert state towards calm and recovery.
Makes sense.
Furthermore, there's evidence it might modulate key enzymes involved in the signaling pathways of other major neurotransmitters, like acetylcholine and the catecholamines, which include dopamine and norepinephrine. Again, it seems less about sending a brand new command and more about ensuring that the existing communications, all the background noise and chatter in the system, are somehow filtered, optimized, and maybe biased towards a state of recovery and stability.
And if we look even deeper, inside the cell, at the level of cellular survival itself, you mentioned earlier it affects key internal pathways. Let's make sure the listener grasps the significance of that. What are these MAPCART pathways, and why is it important that DSIP interacts with them?
Okay, yeah. Think of the MAPCART pathway that's mitogen-activated protein kinase, extracellular signal-regulated kinase pathway, as the cell's internal crisis management team. Or maybe its core survival software program.
Okay.
It's a cascade, a series of proteins within the cell that transmit signals from receptors on the cell surface right down into the nucleus where the genes are. When a cell encounters stress, maybe that's a low oxygen environment like in a stroke, maybe physical damage, maybe toxins, extreme heat, this MAPCART pathway gets activated and helps the cell decide what to do.
Decide whether to.
Decide whether to activate programs for proliferation and repair to try and fix the damage, or if the stress is too severe, whether to trigger apoptosis programmed cell death, basically cellular suicide, to prevent further damage to the organism.
Wow, so that's literally a life or death decision point for the individual cell. And if DSIP is interacting with this critical pathway, it suggests it's somehow influencing that decision, helping the cell choose survival.
That's exactly what the research implies, yes. DSIP seems to promote the activation of these MAPCART pathways in a way that encourages cellular resilience and survival, particularly when the cell is faced with some kind of threat or stressful condition.
Okay.
It really underscores this idea of DSIP as a quiet background technician. It's not just managing sleep or hormones, it's working at a fundamental level to ensure the entire network is resilient and the basic cellular machinery is protected from stress. This might also help explain why its effects are often described as mild or modulatory. It's not causing huge, overt changes, but consistently nudging the system towards stability and survival.
The idea that this tiny, simple 9-amino acid structure is involved in something as fundamental as cellular survival, that really brings us to its truly unexpected talents. Section 5 is all about these other observed actions that seem at first glance maybe disconnected from the initial sleep function, but really confirm its potential role as this fundamental protector molecule.
Yes, the evidence here is quite compelling, though often, again, from preclinical models simulating acute damage or stress. Take its neuroprotective properties, for instance.
Neuroprotection protecting brain cells.
Exactly. In animal models designed to mimic brain ischemia, the lack of blood flow and oxygen that happens during a stroke administering DSIP demonstrated a surprising ability to improve the subsequent recovery of motor function. Animals treated with DSIP performed better on movement tasks after the induced stroke.
Okay, that's significant functional recovery. Did it also reduce the physical damage?
Crucially, yes. Studies showed it also significantly reduced the visible cellular damage, the area of necrosis, or dead tissue in the affected brain regions compared to controls.
That is a profound effect.
Yeah.
It suggests that if an injury like a stroke occurs, DSIP might be part of the body's own frontline response team, trying to limit the damage and preserve function.
It points very strongly towards its regulatory and pro-survival functions activating under conditions of acute duress. And beyond just stroke models, we see other hints of its broader calming or stabilizing effect on the nervous system.
Such as?
Well, some preclinical research has shown DSIP possesses mild pain-reducing properties,
Right.
analgesic effects. This suggests a possible role in modulating nociception, the complex process of sensing and processing pain signals.
Interesting. Pain modulation, too.
And in a similar vein, it's shown measurable anticonvulsant effects in some animal models of epilepsy. This hints at a potential ability to help stabilize overly excitable, seizure-prone neural networks.
So whether the insult is chronic pain signals, or the overfiring of neurons in a seizure, or physical damage from lack of oxygen in a stroke, DSIP seems to be consistently trying to bring the overall excitability levels down towards a more stable baseline.
That seems to be the pattern, yes. And we can potentially find a metabolic explanation for how it might achieve some of this resilience right down at the level of the cell's power generation system. There's strong evidence suggesting DSIP actively supports mitochondrial energy production. Remember, the mitochondria are the powerhouses of the cell. They generate the ATP, the energy currency, that dictates whether a cell has the resources to function normally, let alone weather a storm.
Right, and when a cell is under severe stress, like in a stroke scenario with low oxygen, or even during intense activity, the mitochondria are often the first things to struggle or fail, aren't they? Leading to cell death.
Precisely. Mitochondrial dysfunction is a hallmark of many types of cellular stress and injury. This is why DSIP's potential mitochondrial support is so vital. By helping the cell's energy engines function more efficiently, especially under hypoxic low oxygen conditions, it could provide the crucial energy buffer necessary for survival and subsequent recovery. This might be a key mechanism explaining its observed neuroprotective capabilities.
Wow. Okay, this research really starts to connect the dots beautifully. If DSIP helps the cellular engine run better under duress, then its ability to calm the nervous system, buffer stress hormones, and promote deep restorative sleep. It all starts to align as part of a coordinated survival and recovery strategy.
It does paint a very coherent picture of DSIP as a fundamental regulator of resilience. However, and this is important, we absolutely must reiterate the necessary caveat here.
Which is?
Most of these exciting findings, the neuroprotection, the pain modulation, the seizure effects, even the mitochondrial support, are still potent hints, primarily coming from preclinical animal studies or very early small-scale human observations. They represent really unfinished science.
Right, not confirmed clinical applications.
Not by a long shot. While the promise is immense, the robust, large-scale, objective human data required to translate this into reliable clinical applications is still largely missing, which leads us directly into the very practical challenges researchers face when trying to study this elusive molecule.
Exactly. Which brings us neatly to Section 6. Why is DSIP actually so difficult to study properly? For the listener who's interested in the science and maybe sees conflicting reports, what are the practical roadblocks that contribute to these inconsistent results we keep mentioning? Why isn't the picture clear after 50 years?
Well, the difficulties really start with the molecule itself. We mentioned its poor stability earlier. DSIP is highly vulnerable to degradation by those ubiquitous peptidase enzymes.
The enzymes that chew up peptides.
Right. So in a typical experimental setup, if you just inject the raw native DSIP peptide, it breaks down incredibly fast in the bloodstream or tissues. Often, its half-life is measured in just minutes.
Minutes. So it might be gone before it even reaches its target, or before you could even measure an effect.
That's the very real risk, yes. A researcher might think they're measuring the effect of DSIP, but in reality, they might be measuring the effect of, well, nothing, or just the placebo effect, because the active peptide was degraded almost immediately after administration.
So how do researchers get around that?
It forces them to use extreme care in handling and administration. Or, much more effectively now, they often rely on developing and testing stabilized analogs.
Analogs, like slightly modified versions.
Exactly. Chemically modified versions of the original 9 amino acid sequence that are designed to be resistant to those degrading enzymes. These analogs might have a much longer half-life, allowing them to stick around long enough to exert an effect. But of course, synthesizing, purifying, and validating these analogs adds significant layers of complexity and cost to the research. You also have to prove the analog still works like the original DSIP.
Okay, so stability is hurdle number one. What else? You mentioned timing earlier.
Timing is absolutely paramount, especially when you're dealing with something intrinsically linked to sleep and circadian rhythms. Because DSIP seems tied to the body's natural sleep-wake cycle, the timing of administration has to be highly consistent and very mindful of that internal clock.
What happens if you get the timing wrong?
Well, giving a potential sleep regulator or a system stabilizer at the wrong time of day, say, during the biological daytime when the body is naturally geared up for wakefulness and activity, could entirely negate its intended effect. It might even, paradoxically, cause alerting effects or just confuse the system. It requires highly specialized, chronobiologically informed protocols. You can't just give it whenever.
Right. And then there's the challenge of actually measuring the results, especially for something like sleep quality. How do researchers move beyond just asking people, did you sleep well, which is so subjective?
Exactly. Subjective reports, sleep diaries, questionnaires, they're useful, they provide context, but they are simply insufficient for rigorous scientific proof, especially with something as variable as sleep perception.
So what's needed?
To really prove DSIP's efficacy on sleep architecture, researchers must use quantified objective tools. The gold standard is full polysomnography, or PSG.
PSG, that's the overnight sleep study with all the wires.
That's the one. It involves simultaneous measurement of EEG, brain waves, plus eye movements, muscle tone, heart rate, breathing patterns. The EEG data is absolutely critical here, as it provides concrete, measurable evidence of the different sleep stages, including the presence and duration of that key delta wave activity. Without that objective EEG metric, any claims about improving deep sleep remain pretty tenuous.
Okay, objective measurement is key. And then we can't ignore the elephant in the room for any treatment study, but especially sleep. The power of expectation. The placebo effect.
Oh, absolutely critical. Rigorous controls are essential, particularly the use of double-blind, robust placebo designs. In sleep research, the placebo effect is notoriously strong.
Meaning, just think you got something to help you sleep can actually help you sleep.
Precisely. If a participant expects to sleep better because they know they receive something, that expectation alone can significantly improve their subjective feeling of sleep quality, and sometimes even slightly alter objective measures. It can completely confound the data if not properly controlled for. A strong, well-executed, placebo-controlled, double-blind trial, where neither the participant nor the researcher knows who got the real peptide and who got the placebo, is the only reliable way to isolate the true physiological effect of DSIP from the psychological effect of expectation.
Makes sense. And of course, there's the ever-present hurdle in all biomedical research, translating findings from animals to humans. Why do results from rabbits or rats so often fail to deliver the same impact when tested in people?
Yeah, species differences are always a major factor to consider. The detailed physiology of a rabbit or a rat, while useful as a starting model system, is obviously not a perfect proxy for the human system. Things like the exact distribution pathways for the peptide, the specific activity levels of those degrading enzymes we talked about, maybe even subtle differences in the downstream signaling pathways or the precise modulation points for DSIP. All these could vary significantly between species.
The dose or timing that works great in a rat might be totally off for a human.
It could be. Researchers are constantly navigating this minefield of interspecies variables, all of which contribute to the frustrating inconsistency we often see when trying to move promising molecules like DSIP from the lab bench towards clinical reality. It's a tough process.
It definitely sounds like the messy, challenging frontier of science. Okay, so given all these hurdles, the stability issues, the timing, the measurement controls, species differences, where do you see the biggest opportunities now? Where could young scientists potentially make the breakthroughs needed to finally crack the mystery of DSCP wide open?
There are several really key areas where a significant breakthrough would be transformative for the field. First, and arguably foremost, is the hunt for the receptor.
The Holy Grail, as you called it.
It really is. Identifying the true specific receptor or receptors that DSIP primarily interacts with. Finding that lock for the DSIP key would be monumental. It would immediately unlock our understanding of its entire signaling cascade, providing clear molecular targets for potential drug development or further mechanistic studies. That's priority number one.
Okay. And secondly, still working on the molecule itself.
Absolutely. Continuing the work on developing next-generation DSIP analogs is critical. We need variants that have superior stability and much better resistance to those degrading enzymes, and ideally enhanced bioavailability, meaning more of the administered dose actually reaches the target tissues effectively over a sustained period. Better molecules are needed.
And once you potentially have a more stable, effective molecule, you still have to get it to where it needs to go, right? The delivery mechanism becomes the next bottleneck.
Exactly right. Intravenous injection, which is often used in early studies, is invasive and just not practical for widespread or long-term use. So developing innovative, perhaps non-invasive delivery systems is another major area of opportunity.
Like, oh, what kind of systems?
Things like advanced intranasal sprays, designed to potentially bypass the blood-brain barrier more effectively, or perhaps targeted nanoparticle methods, encapsulating the peptide to protect it and deliver it more specifically to certain tissues or cell types. A major technological breakthrough in delivery could make existing or new analogs much more viable clinically.
Makes sense. And finally, assuming we get better molecules and better delivery, applying these new tools to bigger and better trials.
Yes. We desperately need larger-scale, rigorously objective, double-blind mechanistic trials in humans. Trials that don't just rely on subjective sleep questionnaires, but use gold-standard polysomnography to actually measure delta wave induction and changes in sleep architecture.
And looking beyond just sleep.
And definitely moving beyond just sleep. Exploring DSIP's potential clinical applications in validated models of chronic stress disorders, maybe anxiety, perhaps even neurodegenerative conditions where cellular stress is implicated, or metabolic syndromes. Investigating these areas with the new tools could finally reveal its true therapeutic potential, if any, as a powerful systemic stabilizer and resilience enhancer. A lot of work still to do.
Wow. This has been an incredibly detailed look into what seems to be fundamentally a piece of molecular poetry. I mean, a tiny 9-amino acid structure with potentially such profound system-wide stabilizing effects. Let's try to summarize the key takeaways we really need the listener to hold on to from this deep dive.
Okay. I think the core message is that DSIP is this fascinating, remarkably small nonapeptide. It appears to act as a fundamental regulator, maybe not a driver, but a regulator of deep restorative sleep. But its role extends much further. Despite the ongoing significant mysteries regarding its origin, where it comes from, likely some kind of stress-induced byproduct formed by cleaving a larger protein and despite the lack of a clearly identified dedicated receptor. We have compelling evidence that it effectively acts as a crucial system buffer.
How so?
It seems to help reestablish hormonal equilibrium, partly by blunting the stress hormone ACTH and possibly stimulating restorative hormones like growth hormone. And critically, it appears to provide cellular resilience and even neuroprotection, potentially by supporting mitochondrial function, especially during times of stress or injury. It's really a powerful window into the body's own sophisticated built-in recovery mechanism.
It really is a compelling piece of science, showing us the incredible power of subtle modulation in biology. And before we close, we absolutely need to leave you, the listener, with an important practical note. This comes directly from the source material regarding the current status of this kind of science.
Yes, this is crucial for anyone hearing about peptides like DSIP and getting interested. It is vital for you, as an informed listener, to understand that these types of peptides, including DSIP, are currently distributed strictly for research purposes only. There are chemical regions for laboratory investigation.
Meaning not for personal use.
Meaning they are absolutely not approved for, nor are they intended for, human consumption or self-administration outside of rigorously controlled, ethically approved, regulated clinical trials conducted by qualified researchers.
And why is that distinction so important?
It's vital because of all the challenges we just discussed. The stability issues mean you don't know what dose you're actually getting. The lack of comprehensive, long-term toxicity data in humans means the risks are unknown. Uncontrolled dosing could have unexpected or harmful effects. And the potential interactions with other systems are not fully mapped out. DSIP remains firmly in the basic research and early development phase. We are still studying its potential. We are absolutely not confirming its safety or efficacy for the general public to use at this stage.
Okay, that's a critical point of caution. Thank you. And that leaves us with this final thought for you to maybe mull over. DSIP, as we've heard, seems to act like a whisper in the brain and body. It doesn't shout a primary loud message like some hormones do. It just subtly fine-tunes the entire system. The hormones, the cell survival software, the sleep rhythms working quietly in the background to ensure resilience and stability. So the thought is this. If such a simple, almost hidden molecule can have such profound stabilizing effects across so many diverse and critical systems, what else is your body producing right now? What other subtle whispers are happening right under the surface of our current awareness that we just haven't yet learned how to listen to or understand?
It certainly makes you wonder what other regulatory secrets the body holds. Keep exploring the science of your own inner systems.
Thank you so much for joining us for this deep dive. We'll see you next time.